Um, Good morning, everyone. I've got to make a confession. This is going to be quite green, and it's not going to be about space, but what we can do on Earth to help our very first speakers and the speaker just before me. I only met one of the organizers, Joshua, 3 weeks ago, and this is what happens when you've had a dream of being an astronaut when you're a child, and you're asked, do you mind giving this talk at this conference? And you say yes. So just a couple of disclosure, because I'll be talking about some drugs. It doesn't fund what I'm going to present today. The team that has been working with me is a group of young scientists, and we are very fortunate to be collaborating with a number of clinicians across Australia. What we create is tissue-resident adult stem cell organoids, which I will endeavor to tell you about them today and introduce you to them. These are patient-derived miniaturized organs, which we create in the lab from a patient's stem cell. They have many different applications, and today I will tell you about a few of those. Majority of the samples that I receive in the lab are from patients with a very rare disease called cystic fibrosis. These patients have multiple organ failures, including the lung, liver, and the gut. And it's important because these patients' organs is under constant stress. And I want you to think about, when we talk about space, what that means for us. We are going to be under constant stress. So because this disease is a multi-organ disease, we've been creating miniaturized versions of multiple organs. In 2017, I started the lab, and I started with what I thought was going to be easy, miniaturized version of the airway epithelium, the nose, the lung, and then the gut. And since then, we have explored further into creating mini livers, mini stomachs, and this year we've started creating mini ovaries, which are quite difficult to create. Because this is a rare disease, the type of work that we do is quite collaborative. We work internationally and nationally, and we explore many different applications of organoid biology with the aim of translational research and being able to take drugs or companion diagnostic into clinic to our patients. So what are organoids? They are a lot more sophisticated than the 2D cell cultures that you might be familiar with. These are the ones that we would usually have in the lab, the cancer cell lines, the primary cell lines. They are not as sophisticated as bioprinting. However, these are three-dimensional organoids, which means that the stem cell actually expands and differentiates and creates multiple cell types. For the purpose of this talk, I am not talking about embryonic stem cells, nor am I talking about induced pluripotent stem cells. Rather, I'm talking about stem cells, which are from the adult tissue. So we take a tissue biopsy, isolate stem cells, and from those stem cells, we regenerate and create organoids. I will be talking about the airway organoids and gut organoids briefly. Inside our body, the airway epithelium is stratified. This means that there is a basal layer where there is a stem cell which regenerate, and this stem cell then differentiate into multiple cell types, and these cell types have a specific function. Some of these, such as the ciliated cells, the hair-like structure that beats, and it moves away the mucus, which is created by goblet cells, and the pathogens. Similarly, the gut is also stratified, and there are stem cells that are at the base of the gut crypt, and they create all of these wonderful specialized cells. What in the lab? We try to recreate these developmental pathways from a stem cell into a miniaturized organ. So we brush the nose, or we take a biopsy of the lung, or maybe the gut, and then we introduce these stem cells that we have isolated into certain conditions that allows this stem cell to generate more and more and more of it. We need lots of these stem cells to be able to biobank them, so we can take them out and test many, many drugs on them. Once we have enough of these stem cells, then we start differentiating. These are cues that we provide to the stem cells for the stem cell to actually start differentiating into all those multiple cell types that I mentioned. We can do this under a number of different conditions. They are called either being present on a porous membrane, air-liquid interface, or we embed them in an extracellular matrix. The type of organoid that are generated are quite different. However, what this allows us to do is to mimic that miniaturized lung and then do a lot of different functional assays on them. So here's an example of when you take a stem cell and you just culture it on a plastic dish, or when you take it and you culture it on the porous membrane. That stem cells after nearly 30 days will create a miniaturized lung. And that miniaturized lung is functional. Over here, you can see these are the tiny hair-like cilia structures and the cilia beads. And what we do in the lab, this is the functional assay I was mentioning, we can actually measure how fast the cilia beats, we can measure how coordinated the cilia beats. And this measure of frequency and coordination changes depending on if you have a patient with a certain disease or if you are introducing a drug here, or if you're exposing the organoids to microgravity. Here we have the organoids in a matrigel compartment. This allows you to have, if you can imagine, like a small alveoli. So you've got these sphere structures where the inside of the organoid is hollow, filled with mucus. And this is important for us because not only we can measure the cilia again in this model, but we can actually test different drugs and understand how these drugs might alter the ion channels in the lung. You would ask me, Shafagh, how do you know you've got all of the different cell types? Well, we can actually explore this by RNA sequencing and have a look at deconvoluting the transcriptome to find out that, yes, we absolutely have all of the different cell types, which is in our lung here in the lab in these models. We do very similarly with the gut, which I won't bore you about, but also the gut produces these functional mini organs for you. This is what the lab has been working on since 2017. We've got a biobank of nearly 200 patients with CF and around 50 individuals, which are healthy controls, and a couple of other respiratory diseases. What I would like you to pay attention to is the type of functional assays that we have developed. So it's quite important to be able to create these things. As mentioned earlier, we might be able to bioprint these, and we might be able to transplant and have skin grafts, or even go fancier and transplant into a dysfunctional lung. However, we need to make sure that we actually are producing the correct model, and also to be able to characterize them. So my lab has been working towards 3 functional assays. One of them, the very first one, is to be able to measure ion channel function. This is important because it allows us to know how ion moves, which is as a result of different drugs that we've been working with. The other assay that we look at is a forskolin-induced swelling assay. This is a fluid movement. And the third one is that cilia beat frequency that I mentioned. And over here is just an example of how the functional assay works. So this is a healthy individual with functioning CFTR channels, which is a chloride channel. Here is a patient with CF, which doesn't have a functioning CF channel, has been provided a drug, but is non-responsive. And over here is a different CF patient, has been provided a drug, and their gut organoid is fixed as a result of this drug, and as such is responding. This is primarily what my lab does. We look at patients with rare type of mutation, and we try and customize different treatments for them. For the purpose of today, I want to tell you about a side project of the lab, which is looking at hypoxia, which is lack of oxygen, and how that lack of oxygen, and whenever I say lack of oxygen, think about, she's talking about microgravity. What happens if stem cells are grown in different oxygen concentration? So this is what the lab did. We took the stem cells from the nose of 11 donors. We divided these stem cells to grow them under normal oxygen level and dropped the oxygen level down to 2%. And after we expanded the stem cells, then we differentiated them. This differentiation happened under 2 different oxygen conditions. However, we did a crossover. So the normoxic stem cells were cultured in hypoxia to differentiate. The hypoxic stem cells were then cultured in normoxia and vice versa. So 4 differentiation conditions. What we noticed, this is at the stem cell level, as you would expect, there wasn't many changes in the shape of the cells, nor was there a difference in the way that the cells were actually dividing. However, at the proteome level, we could see major differences between the proteome of hypoxic and normoxic stem cells. One of the major differences that we could see was inflammation, and that there was a reduction in inflammation in the hypoxic environment, although oxidative stress was going up. And over here in green, you can see there is a whole lot of H1F, which is a hypoxic protein present. So this is what's happening at the stem cell. What happens when cells are differentiated? I don't want you to worry about this very busy proteomics plot over here. But what was quite interesting for us was the fact that there were a couple of conditions where the proteome did not change. However, there were conditions where the proteome was most severely affected. When I think about this in the context of space, this makes me wonder whether we need preconditioning before going into the space. Because what I can see here is that when the stem cells were in the normoxic or the hypoxic environment, and then we cross them over, that's when the stress goes up. However, whether they were in the normoxic or the hypoxic and continued in that environment, they seem to be dealing a lot better with the stressor that was given to them. The pathways that were coming up were those that we had expected. What we could notice quite severely is that the thickness of the lung epithelium was reducing quite dramatically when we were differentiating in hypoxia. This is something that I discussed with Joshua quite a bit as to what would happen if we actually differentiate our stem cells in microgravity. Are we going to end up with much thinner lung epithelium? And what would be the consequence of that for our space biology? What we could also see is that not only we have a thinner epithelium, but that the tight junctions that puts these cells together are not working as well. There is a reduction in that as well. And that's what you see over here. And it didn't matter. As long as you were differentiating in hypoxia, you had a drop in resistance. We could see gross changes with differentiation in hypoxia. Your lungs usually have a good balance of cilia and mucus cells. What we could see was that when the cells were differentiated in normoxia, the normal condition, we had that good balance. However, those stem cells that were differentiated in hypoxia had shifted quite significantly towards a mucus-producing cell type. So a lot more mucus was being present in that really thin epithelium. This is quite significant when you think about the effect of it if we are trying to breathe. Interestingly, the cilia of these hair-like structures, they had shrunk quite significantly when the stem cell was differentiating under hypoxia. Not only they had shrunk quite significantly, the beating of these cilia was a lot slower. Their coordination was a lot better. And the reason it coordinates better is because it's beating slow. It doesn't necessarily mean it's a good thing. We had an overproduction of inflammatory compounds such as IL-8 when cells are differentiated under hypoxia. So you can imagine we are dealing with an inflamed state. And when we test different medication on these different cell types now, what we see is that the response to medication was actually quite different. Under hypoxia, some of these medications, instead of producing the response that we anticipated, which is around this 35 microamp, had significantly dropped. And what that tells me is that should the patient receive this medication, they would not be responding to the medication as effectively. So with this in mind, I guess what I want to say is what would be the impact of microgravity on stem cell when we expand the stem cell and we differentiate them. So unlike trying to do a very short-term experiment of a 1 hour, 2 hour of a stem cell or an organoid on the microgravity, what I would like to be able to achieve, should it be possible to create the devices in a way that would allow us to do a chronic and continuous microgravity exposure. The organoids takes between 1 month to a month and a half to expand and differentiate. And as a result, it will require to be able to have a group of scientists and engineers who would be able to change the current microgravity devices to accommodate this. Thank you so much.